Morning, everyone. Thanks for being here. I'm very happy to share with you some of the results um, of the work that we've been doing in the myeloma realm with regard to COVID-19. And the work that I'm going to talk about in this very brief talk is currently under review and was titled A Tertiary Center Experience of Multiple Myeloma Patients with COVID-19. Lessons learned and the path forward. So the objectives of what we'd like to go over today is to explore the myeloma population at Mount Sinai that developed COVID-19 and also to analyze some of the risk factors associated with poor outcomes in this myeloma cohort. So I will briefly take you through a background and study design, some demographics of the population that we explored, disease characteristics, and also risk factors for hospitalization and mortality, and also as a little added bonus at the end, antibody response information that we've acquired so far in our patients. So COVID-19, as everyone knows and has personally experienced, is caused by the SARS-CoV-2 virus, and it definitely very drastically altered how we deliver healthcare, not only in the nation but particularly in New York City, and especially at our cancer center where we saw so many patients. Just in New York alone, by May 22nd, there were that's when we submitted, we're moving towards submitting the paper. There were over 200,000 cases already registered and confirmed in New York and 16,000 deaths, with an overall death rate based on most literature about 21% for those confirmed positive. And of all the deaths, about 8% were patients who had cancer. And so why did we ask this question here at our cancer center? Well, we have over 3,000 active cancer myeloma patients that we see at Mount Sinai Hospital. We have a highly diverse patient base, which is also reflected by our cancer center's general population. We have very medically complicated patients, some of whom are on many lines of on their nth line of myeloma therapy. And one particular unique thing about our myeloma patients is that they suffer from what is both a cellular and a humoral type of immune dysfunction, something we call immunoparesis. So essentially, if a patient who has IgG myeloma, for example, has either a IgA or an IgM that is subpar, or both, that is basically defined as what we call immunoparesis. And a large population of myeloma patients, including the patients that we see, are immunoparese. And what this represents is an at-risk immune system to many infections, including what we think would be COVID. So the study design is we early on created a database of patients who and kept track of a database of patients who developed COVID-19, both who were tested at our system and also at external systems, external health centers. And the patients who developed COVID-19 between March 1 and April 30th were included on this publication that we submitted. And there were a total of 58 patients who were confirmed to be COVID positive. 36 ended up hospitalized overall at some point, and 22 remained fully outpatient for the duration of their COVID illness. And 54 were multiple myeloma, while 4 were smoldering myeloma. So in the interest of time, limited time, I'm going to take you through some of the more interesting parts of the exploration that we performed in our 58 patient population. Just the overall age, none of this is too surprising, but overall age is about 67 for myeloma, which tends to happen in an older population. And if you look at the sex of overall patients that were hospitalized, you'll see that there was quite a lean toward male patients that ended up coming into either our hospital or an external hospital for COVID at 68%, or 57% for the ones who ended up unfortunately not surviving. And one interesting tidbit here on the right side also is that non-white patients in this N of 58 population, so keep in mind limited population, but 92% of the ones who ultimately did not survive COVID were non-white background. And in terms of the ones in terms of obesity, people with greater than 30 BMI, there was a higher percentage of patients who died who were actually obese. Also, as we may have seen in other studies as well, cardiovascular risk factors seem to be more pronounced in patients who ended up being hospitalized. In terms of disease characteristics, across the board, there's a lot of data here, but the point really is that whether you remained outpatient or were hospitalized or were hospitalized and did not survive, for the most part, you see very similar data across the board here that disease background was not really associated with any of those outcomes. And also patients, regardless of whatever treatment patients were on coming in with at the time that they came in with COVID, there was no real difference, especially statistically, there was no difference in terms of their outcomes, the associated outcome. And if you look at the bottom here, the bottom portion of this slide, these represent labs that were performed at the last outpatient visit. The one thing that I'd like to highlight here is that patients who had severe hypogammaglobulinemia, which is defined as IgG less than 400 milligrams per deciliter, these patients tended to be associated more with actually statistically associated with worse mortality in this patient cohort. As you can see, about 89%, so pretty much most of the patients, large majority, were what we kind of consider classically as at risk from an immune perspective. And in terms of biochemical markers, so this subset here is actually N of 23, which means that this represents patients that were actually hospitalized at our main hospital, Mount Sinai Hospital, where we had full biochemical data reporting because patients had their labs drawn, for example. So if you look at this population for closer analysis, if you look at the peak what we consider classic inflammatory markers, CRP, ferritin, D-dimer, if you had worse values during your hospitalization, these were actually associated with, we saw a statistical association with increased mortality. The striking one here that you can see on the page is the D-dimer. Patients who survived versus who did not, for example, 2 versus 20, that's a 10-fold difference and was very profound. Something that we can, just as an added bonus here, we followed these classic inflammatory markers over time, and we also followed the cytokines as well. But we included here is that CRP, D-dimer, ferritin, and LDH, as you can see, red here on these curves means that each red line represents a patient that did not survive, and a blue line represents a patient that did survive. So for D-dimer, there's quite a striking separation in the direction of their overall regression line, and also of the between the red colored one on the top right. You can see that it was clearly associated with worse D-dimer was associated with worse survival, and a D-dimer that may have peaked but then came back down in our patients, at least at Mount Sinai Hospital. You can see a nice flattening out eventually of their D-dimer for the patients who ended up surviving. So just to kind of highlight statistically the associations that we found to be significant for hospitalization and also for increased mortality. In terms of factors that were associated with a higher hospitalization rate, that included age greater than 70 years, a male sex, a history of CAD or CHF, and in general, high cardiovascular risk profile, diabetes. Also, the number of comorbidities that the patient had coming in when they developed COVID, and also pre-existing lymphopenia, less than 500, 0.5. And factors associated with increased mortality we found to be non-white race in this population, and statin use, and also a higher peak levels of the of our classic inflammatory biomarkers, and also procalcitonin, which is not a classic inflammatory biomarker. It's associated with bacterial infection. In terms of antibody response, earlier this month when we looked, 96% of our patients, keep in mind 89% immunoporeced patients, so not optimal humoral immunity by definition. 96% of patients actually had developed antibodies to SARS-CoV-2 at a median time of about 32 days after their initial diagnosis with COVID. The titers range from a low titer, 1 to 160, up to the max titer is 2880. And more than 2/3 had actually developed the highest level titers possible. So we're not really sure, I guess. This is looking forward a little bit. We're not exactly sure in the general population if having high antibodies is directly correlated with prevention of reinfection, but that's something we certainly will keep in mind, and we'll continue to follow in this population, I think, because that would be really interesting. And also, we were able to track the median time to PCR negativity when we re-swabbed these patients before any subsequent treatments once they had resolved the initial infections. And it took about 43 days for patients to test negative again, with a range of 19 to 68 days. And just to finally, there are more studies than this actually ongoing with cancer and COVID-19, but these are, I've included the ones that are specifically looking at myeloma. And as you can see, we're on the bottom here, but most of the other studies have pretty immunoporeced population in this red box here, 86%, 90, and then we're 89%. Death rates seem to vary in the various cohorts that have been sent for publication so far, with higher than 50% in this British one in number 2, whereas a German one, the top two are multi-center. The British one, the German one at the top, 21 patients, they claimed a death rate of 0%. And we are at about 24%, which is consistent with what we're seeing in the New York area. We're eagerly also awaiting our own larger cancer center study here that Dr. Smith and Dr. Kaita are working on. And so, in summary, New York Center, unfortunately, was at the epicenter of the COVID-19 pandemic. And we think there's definitely conflicting risk profile for delaying myeloma treatment and infection risk in vulnerable patients during a time, especially of pandemic. The mortality rate that we saw in our cohort was pretty similar to overall in-hospital death in New York City, with 24% in our cohort. And also, importantly, actually, the myeloma-specific disease characteristics, none of them were significantly associated with mortality of any kind. So what that really tells us, I think, is that, of course, at some point there may be outbreaks, there may be, there may be maybe second waves, quote, quote. And what we think is that it's important, of course, to maintain early diagnosis and promptly intervene when a patient is confirmed positive, of course. But there does not seem to be a need to preemptively change and plan a patient's entire treatment course for myeloma. And it's still very important to treat the myeloma up until the point where a patient is suspected to have COVID and then needs to be tested. So in terms of, I mentioned this briefly, antibody development, we're going to have to see if this is protective, but this is very promising so far to see that almost all our patients that have been tested have developed antibodies. So this database will be continued. We'll keep track and we'll add more patients, and hopefully there will be a part 2 that we can discuss at some point in the future. I'd like to thank the entire myeloma team, Dr. Maduri, who worked very closely with us, and also some of our other team members. And also thank you to Dr. Kozak, Dr. Malone, and Wanda, and all the co-fellows for the support so far, and also our collaborators in the immune monitoring core, Diane, Sasha, and Miriam. I'll take any questions.
Very nice. So I don't think we have a lot of time for questions, but quickly, while Doug is pulling up his slides, could I ask you therapies? Did the therapy matter that the patients were receiving?
Did you?
The myeloma therapy. The myeloma therapy.
No, it did not matter. So, daratumumab, for example, the CD38s or IMiDs, none of that was directly associated with any outcome.
Thanks.
I can pull up my slides if Bo, you can stop sharing. I can.
Sorry, Doug, let me do that. I forgot to unshare. How do I unshare? I'll leave meeting. I'll come back.
It should say.
Oh, there you go. Okay. Can everyone see my screen?
Yes, we can.
Okay, great. So I'll hurry through this as to not run over time. This is going to be a kind of preclinical rationale for a phase 1/1b study of ONC201, which is an exciting agent for myelofibrosis that utilizes the apoptotic pathway. So I'll go through the treatment landscape, describe these apoptotic pathway, and then describe the trial, which is currently running through the regulatory framework. So John Feld showed a similar slide last week, and I thought it was really important to give some context for what the therapeutic landscape of myelofibrosis is. There's only 2 approved therapies for myelofibrosis: Jakafi, ruxolitinib, and fedratinib. And both of them are primarily work as JAK inhibitors, which takes advantage of a central pathophysiologic pathway in myelofibrosis with overactivation of the JAK-STAT pathway, leading to downstream effectors, which leads to changes in the bone marrow and cytokine release that causes a lot of the symptoms that are so debilitating for patients with myelofibrosis. And ruxolitinib showed its efficacy in the COMFORT 1 and 2 studies, demonstrating really excellent spleen responses and symptom responses. And fedratinib in the Jakarta 1 study showed evidence that it can really improve spleen responses compared to placebo, and as a second-line agent in the Jakarta 2 study showed evidence that it can reduce spleen volumes even after patients were treated with ruxolitinib. And while there is a survival benefit that most people in the field believe occurs, it is pretty well established that ruxolitinib and most likely fedratinib does not change the natural disease process of myelofibrosis, which is progression to leukemia. So clearly the field needs to move on to pathways that really target the stem cell and the pathobiology to really change the disease course of myelofibrosis. And also concerning is that patients who fail ruxolitinib either through intolerance or they come off because they didn't have an adequate response have a pretty dismal survival. So this is a large database study from a number of US-based databases that shows that the overall survival of patients who are on ruxolitinib after they stop ruxolitinib is only 11 months. And this is in line with other studies from Moffitt and from MD Anderson showing a poor survival. And I also want to bring your attention to the time to next treatment, which was not estimatable in this study, showing that even patients who fail ruxolitinib, a lot of them never even get to go on to a second-line therapy. So clearly, we're looking for other agents that are urgently needed for this very high-risk group that can also affect the underlying pathobiology of myelofibrosis. So one such pathway that's been exploited in a lot of hematologic malignancies with great success is apoptosis. So roughly, this is broken up into an intrinsic and extrinsic pathway. And the extrinsic pathway starts with TRAIL, TNF-related apoptosis-inducing ligand. And that binds to certain death receptors such as DR4 or DR5 and results in oligomerization of other proteins forming space, and space moves through cleave BID to the pro-apoptotic proteins BAX and BAC. And BAX to BAC results in mitochondrial membrane permeation and release of cytochrome C and Diablo, which is just a really cool name for an apoptotic protein. Cytochrome C combines with other factors to cleave caspase 3 that really results in apoptosis, and also causes more activated caspase 8, thereby further amplifying the apoptotic pathway. So ONC201 was designed to target and increase the activity and expression of TRAIL. So it started with a screen by Joshua Allen, and prioritization assays were used until from the TCI chemical library to find compounds that could induce TRAIL on tumor cells. And so it came up with ONC201, and in its mechanism, it leads to inactivation of RAS effectors, AKT and ERK, through DRD2, dopamine receptor D2, and through FOXO3A leads to increased expression of TRAIL. It also has another mechanism where it can activate caseolinic protein P, which leads to mitochondrial dysfunction and apoptosis. So multiple mechanisms that causes apoptosis in cancer cells. But really interesting for us in the myelofibrosis field is that it has been shown in a cutaneous T-cell lymphoma line to also downregulate the JAK-STAT pathway, as shown in this study. And so it really seems like a really amazing candidate compound for myelofibrosis. It's been investigated in a number of other malignancies, such as glioblastoma, and both in company-sponsored and investigator-initiated, and include a myeloma study, which was run through our myeloma center here. But it's not really well known what effects ONC201 has on myelofibrosis cells. So this is work from Min Liu in Dr. Hoffman's lab. It's really elegant work that shows that in hematopoietic samples taken, CD34 positive samples taken from MF patients, there's a decreased amount of DR4 and trail as compared to normal controls. And in addition, DRD2 and DRD3, which are the receptors for ONC201, are also upregulated in myelofibrosis cells, both in the peripheral blood and in the spleen. So this comes from an abstract from last year's ASH that's really elegant work that shows combining ONC201, which targets the extrinsic apoptotic pathway, with RG712, which is MDM2 inhibitor, a class of drugs called nutlins, not only increases trail expression individually, but also in combination. And whenever Min looked at colony numbers of hematopoietic colony cells of normal patients, there was no real effect in the number of colonies with increased amounts of ONC201. But in patients with samples from patients with myelofibrosis, there was a dose-dependent decrease in the number of colonies with ONC201 that was also further potentiated if added in with the nutlin. So given this really compelling preclinical rationale and experiments done by Min on MF primary cells, we devised a phase 1/1b clinical trial to evaluate ONC201 in the clinic. So the phase 1 portion is going to target patients, this high-risk group of patients with a poor overall survival who have failed ruxolitinib. So have been on a JAK inhibitor for greater than 3 months and had an inadequate spleen response. And they'll be getting ONC201 weekly and showing a response assessment after 7 cycles. But the phase 1b portion is where we're really going to evaluate its efficacy. And in an expansion cohort of ONC201 at the recommended phase 2 dose, we'll be giving 9 patients with myelofibrosis who have also failed the JAK inhibitor ONC201 for response. But we're also going to evaluate ONC201 in combination with ruxolitinib to see if this combination is both safe and see if that we can improve upon the responses that are seen with ruxolitinib. So the primary objective in the phase 1 will be to estimate the maximally tolerated dose, and in phase 1b to estimate the efficacy. And secondary objectives will be response by IWG ELN criteria and adverse events. But we're also really interested in some exploratory outcomes of symptoms, but also a lot of really great correlative work through Min and Dr. Hoffman, looking at protein levels of trail, caspase cleave, cytokeratin 18A. And we'll also be looking at the pharmacokinetics of ONC201. So this trial is now near completion of being written and is now going through the regulatory steps necessary to open it. The phase 1 will be open here at Mount Sinai, with the phase 1b planning to be expanded across the Myeloproliferative Research Consortium. And we have agreement with Oncoceutics, who makes ONC201, to provide drug and have supported the study. So we're short on time, but I just wanted to acknowledge my mentors, Dr. Mascarenas and Dr. Hoffman, as well as Dr. Liu, for all that great preclinical work, as well as Lynette from the regulatory standpoint, Heidi, who's a biostatistician for the MPNRC, as well as my program directors and co-fellows. And with that, I think I have 2 minutes if anyone has any questions. Thank you.
Thanks for finishing on time, Doug. Any toxicity? Did you go over the toxicity? I don't know if you mentioned.
So it's in the weekly dosing with other tumor types, it's extremely well tolerated. If you increase the dosing, has been done in an investigator-initiated trial in AML. There was reports of some toxicity seen, but in general, it is extremely well tolerated drug. and we expect to move through the phase 1 portion without any DLTs fairly rapidly.
Doug, Doug, it's Ron. Maybe just to put it in perspective, you could give them a little summary of the data in glioblastoma, which is very promising.
Yeah, so it's a candidate compound for glioblastoma for a number of reasons, both that it can cross the blood-brain barrier, but also there's, because of the DRD2 receptor, it seems like this might be a particularly potent compound. And there's been at least 2 or 3 phase 2 trials that have evaluated ONC201 and showed objective responses, and particularly with certain mutational subtypes. And that is while having virtually no toxicity with the drug. So it's being kind of in a number of different types.
Okay, thanks,
Great.
Doug. I
Thanks
appreciate
so
it.
much.
Excellent talk.